this video, we are going to discuss the structure of DNA and the awesome process in which is called replication, which is kind of like something from an X-Men film, but in real life.
The double helix is a description of the structure of a DNA molecule. A DNA molecule consists of two strands that wind around each other like a twisted ladder. Each strand has a backbone made of alternating groups of sugar, which is deoxyribose, and phosphate groups.
Since we're using an awesome Connects model, let me show you what these pieces represent. The gray piece represents the deoxyribose sugar. The orange piece represents 2 hydrogen bonds. The brown piece represents 3 hydrogen bonds. The purple piece represents a phosphate group. The gray with the green represents cytosine and guanine, and the black and the white represents adenine and thymine bonded together. Watson and Crick discovered that DNA has 2 sides, or strands, and that these strands were twisted together like a twisted ladder, the double helix. The sides of the ladder comprise the sugar-phosphate portions of adjacent nucleotides bonded together. The phosphate of one nucleotide is covalently bound to the sugar of the next nucleotide. The hydrogen bonds between phosphates cause the DNA strand to twist. The nitrogenous bases point inward on the ladder and form pairs with bases on the other side like rungs. Each base pair is formed from 2 complementary nucleotides, bound together by hydrogen bonds. The base pair in DNA are adenine with thymine and cytosine with guanine.
The orange piece right here represents the 2 H bonds that are between thymine and adenine, and the brown piece in the middle right here represents the 3 H bonds between guanine and cytosine.
Here we have a snippet of the DNA after it has finished replication. On the left here, we see that this is the daughter strand. On the right, the parent strand. The sugars are represented here by gray and yellow. The bonds between guanine and cytosine are 3 hydrogen bonds, whereas between thymine and adenine, we have 2 hydrogen bonds. And here the purple represents the phosphate groups. As we discussed before, DNA sequence runs from 5' to 3' on the top strand and 3' to 5' on the bottom strand.
Now that we have fully explained to you all the components of this Connects DNA structure, what exactly is DNA? DNA is short for deoxyribonucleic acid, and it's a molecule that encodes the genetic instructions used in the development and functioning of all living organisms and even viruses.
We are now going to talk about DNA replication.
Semi-conservative replication describes the mechanism by which DNA is replicated in all known cells. This mechanism of replication was one of 3 models originally proposed for DNA replication. Semi-conservative replication would produce 2 copies that each contained one of the original strands and 1 new strand.
DNA replication begins with DNA helicase splitting the gene in half. This is followed by DNA polymerase, which matches the base pairs of the parent with those of the daughter. This creates a semi-conservative strand, which consists of parent and the newly formed daughter strand.
After replication is done, this is the end product of what DNA would look like after the process. As you can see here, the silver parts, which are right here, that's the parent half, and the yellow half is the daughter strand.
10 interesting facts about DNA. 1. Even though it codes for all the information that makes up an organism, DNA is built using only 4 building blocks, the nucleotides adenine, guanine, thymine, and cytosine. 2. Every human being shares 99% of their DNA with every other human. 3. If you put all the DNA molecules in your body, end to end, the DNA would reach from the Earth to the Sun and back over 600 times. 4. A parent and child share 99.5% of the same DNA. 5. If you have 98% of your DNA in common with a chimpanzee. 6. If you could type 60 words per minute, 8 hours a day, it would take approximately 50 years to type the human genome. 7. DNA is a fragile molecule. About 1,000 times a day, something happens to it that causes errors. This could include errors during transcription, damage from ultraviolet light, or any host of other activities. There are many repair mechanisms, but some damage isn't repaired. This means that you can carry mutations. Some of the mutations cause no harm, a few are helpful, while others can cause disease such as cancer. 8. A scientist at Cambridge University believes humans have DNA in common with the mudworm, and that is the closest intervertebrate genetic relative to us. In other words, you have more in common, genetically speaking, with a mudworm than you do with a spider or octopus or cockroach. 9. Humans and cabbage share about 40 to 50% common DNA. 10. Frederick Miescher discovered DNA in 1869. Although scientists did not understand DNA was the genetic material in cells until 1943. Prior to this time, it was widely believed that proteins stored genetic information.
And that, ladies and gentlemen,
is the end!